Kia ora and good afternoon, and welcome to the LENScience Senior Biology Seminar Series 2010. We're at Seminar 7 today, which means only one more seminar to go in the seminar series, and we hope that you have really been enjoying everything that we've had to offer to you. Today I'm joined by Professor Lisa Mattisou-Smith, who is one of the principal investigators with the Alan Wilson Centre and also Professor of Biological Anthropology at Otago University. And together we're going to be talking to you about human settlement of the Pacific, in particular looking at how recent evidence collected from molecular biotechnologies is helping us to rethink the origins of modern Polynesians. Now, before we get into that, just a little bit about the Alan Wilson Centre. This is the second seminar that we've actually done featuring researchers from the Alan Wilson Centre. But a quick reminder for you, Alan Wilson was a very top scientist in New Zealand, well renowned overseas for his work in human evolution, and particularly in the use of technology to understand human evolution. Now, some of you may have heard the phrase mitochondrial Eve, and it was the work of Alan Wilson and fellow researchers that helped develop this model of mitochondrial Eve, which supports the out of Africa hypothesis for the migration of humans out of Africa. And we're going to talk a little bit more about out of Africa a little bit later on in the seminar. But before we do that, it's been a bit of a break since the last seminar, and so some of you have probably forgotten about the wiki discussion that was going on. If you actually haven't been in and had a look at some of the quality of some of the answers, I strongly recommend that you do so. The work that has been done by students like you from around the country and here in the live audience is very, very good. And it's making choosing somebody to give the prize to each week getting harder and harder. So it's my pleasure to actually say the prize for the best discussion last week goes to Tom from Riccarton High School. Now, I chose Tom's as the winner of this prize because his answer to the challenge question was actually extremely well structured, very logical and very well thought out. And what he did is he developed his answer. Now, when you're working in scholarship, that is something that the examiner often comments on, is that the top students who are getting scholarship and scholarship without outstanding performance are able to think logically about how they approach the question and structuring their answer to it. And that's the reason why I picked on Tom as a very good example of this. If you haven't already been in and have had a look at his answer, I suggest that you do so. So we're looking today at human settlement of the Pacific. As we talk through the seminar, I'd like you to be thinking about these concepts which you have covered in your Year 13 biology course this year, and think about how they apply to what you are hearing, because that's what scholarship is going to require you to do, and not very far away. When you are analysing the question presented to you in scholarship, part of that analysis is thinking, what are the biological concepts that I have learnt about that this is illustrating, that are underpinning the situation presented to me in that question? And when you've done the analysis, it's about pulling together ideas from the question, ideas from what you've learnt in class, integrating them together into coherent response that the markers can see your thinking and your understanding. Now, I'm sure by now you're going to be able to tell me what the achievement standards are that link to this particular seminar. So there they are up on the screen just for you to do your little mental check. So we're talking about human migration in the Pacific. And I want you to just stop and think a moment. We know that Polynesian peoples are amongst the best navigators in the world. They actually explored and settled the Pacific well before Europeans arrived in their big ships with all their compasses and charts and those sorts of things. The Polynesian people went out there and just used their extensive knowledge and understanding of the natural world, of tides, of stars, of waves, of bird migrations, to help them navigate their way around and to find and explore all the islands that we now know as modern Polynesia. So that's at the heart of what we're talking about there, is this, who were these people? Where did they come from? And the Pacific is actually a really interesting place to be looking at and answering these questions, because not only do we have some of the earliest archaeological remains of that out of Africa migration that I talked about just before, we also have evidence of the last great human migration, which was the migration from island Southeast Asia through into what we call Polynesia now. So this seminar is going to start by linking back to that migration out of Africa. The migration of the first anatomically modern humans, which took place around 100 to 150,000 years ago. Now, the first migration, we know, followed the coastline out of Africa through India and into Asia. That migration then moved down into Australia and arrived there by, at the very latest, 45,000 years before present. And at a similar time, there was another wave of migration north into Europe and up into, north into Europe. A later migration went further up the coast of Asia and crossed the Bering Strait into what we now know as North America. But it's actually that arrow that's going down into island Southeast Asia that we're going to be focusing on today. Who were those people and what happened to them next? And over the course of the seminar, we're going to be answering those questions. But how do we know that data? How have we put that map together? Obviously, a lot of the evidence has come from archaeological remains, but increasingly, modern biotechnologies are helping us to fill out that picture and to make it more complete through studies of DNA. And this is where we're going to do a little bit of a review over some stuff that you would have learnt last year in Year 12 biology. So of course you will recognise that as a cell. And you all know in the middle of the cell there's a nucleus. And you'll be aware that of course cells have mitochondria. They're the powerhouse of the cell, providing the cell with energy. But the mitochondria also has a role to play in helping us understand the story of the human migration into the Pacific. So let's look a little bit more closely at that. First of all, the nucleus contains chromosomes, and those chromosomes contain DNA. And we refer to that as nuclear DNA. But mitochondria also have DNA inside them. Now, the mitochondrial DNA is slightly different from nuclear DNA. It's a lot smaller, only about 16,500 base pairs, and it's found in a ring rather than a long chain like we see in the nuclear DNA. So during the seminar, we're going to be referring to both the nuclear DNA and we're going to be referring to the mitochondrial DNA. Now, why is mitochondrial DNA so interesting when we're coming to work out what is the story of the migration of people into the Pacific? And the answer to that question is to do with how it is inherited. So what we have here in this diagram is a picture of an egg and a sperm. Now, when fertilisation occurs, go back a step, both the egg and the sperm, of course, have nuclei, so they have nuclear DNA inside them. They both also have mitochondria because that provides the energy to keep the cell functioning. Now, when fertilisation occurs, the only thing that actually happens is the nuclear material from the sperm goes into the egg. If you look very closely at the nucleus, you can see that it's now consisting of both the pink and the blue DNA material. So that fertilised egg has got all the mitochondria that was there from the mother, but doesn't have any of the mitochondria from the sperm. It's only nuclear DNA goes through. Now, that fertilised egg is now going to go through a process of cell division and specialisation until we end up looking like we do now. And what this means is that every cell inside your body and my body, the mitochondria in that was inherited from our mother. It hasn't gone through any recombination or any crossing over or anything like that. So it is virtually identical to the mitochondrial DNA of your mother. There may be one or two mutations, and that's actually what interests the scientists. Because by looking at this mitochondrial DNA, we can actually trace the movement of females around the world. And Lisa's going to show you in a few minutes a little bit more about how we do that. Now, I can hear some of you probably saying, well, that's very well, but females are only half of the human population. What about men? Well, there is actually a way that we can do the same for men as well. And that involves looking at nuclear DNA this time. Now, you've all seen one of these before. It's a karyotype showing the human chromosomes. The 22 pairs of autosomal chromosomes, and then down in the bottom right-hand corner, the sex chromosomes. Now, what men have that women don't, of course, is a Y chromosome. And that Y chromosome can also be used to provide evidence in a similar way to the way that we look at mitochondrial DNA. How can it do that? We need to look at its structure a little bit more. Now, the grey part of that chromosome doesn't go through any recombination. Now, you all remember recombination occurs in meiosis, adjacent chromosomes exchanging genetic material. The Y chromosome as well is passed on to every single male. So that area in the middle we call the non-recombining portion. And that portion allows us to trace male lineages in a similar way that we trace female lineages using mitochondrial DNA. Because like the mitochondrial DNA in females, it's passed on from father to son this time, virtually unaltered. The only changes may be as a result of mutation. So because we know that nothing's changing the mitochondrial DNA and the Y chromosome DNA apart from mutation, then we can use that to look at family trees and then from there look at movement of peoples around the world. How? That's what Lisa's going to tell you.
Now, as Marshall's just explained, we can combine the use of mitochondrial DNA to look at maternal history and Y chromosome to look at paternal history. But one thing we really have to remember, and a lot of people get quite confused about, is when we're looking at mitochondrial DNA or Y chromosome DNA, we're only looking at a portion of your ancestry, a portion of the family tree. So when we look at Y chromosome DNA, and we have you, in this case you being a male, we see that we're tracing the DNA that was inherited from your father, which he got from his father and from his father. So it's looking at that paternal line. Okay? So men are inheriting their Y chromosome through that family line. We can look at the mitochondrial DNA as well, in both men and women. And that mitochondrial DNA is telling us about the maternal history, your mother's 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 line. Okay? So you're combining this, you're getting a double approach, but there are also a whole lot of your relatives, okay, that are as closely related to you as the lines that we're tracking that aren't actually included in that DNA analysis. So all of the people, your mother's father and your father's mother, you're not learning about their history when we are giving you, for example, a mitochondrial DNA or a Y chromosome history for you. So very often people say, when they have done their DNA through any of the websites that you can actually get a DNA kit and send in your own DNA, and they say, well, this actually conflicts with my family's history. The stories that are passed down, that we heard that we were originally related to Charlemagne or to Pocahontas or to somebody, in history, and that's why they're not necessarily inconsistent with one another. The stories that you're getting might be talking about a whole other part of your family that's not actually being traced through the mitochondrial DNA or the Y chromosome. So that's something actually that's really important to remember when you're looking at this kind of information. We're only getting a portion of the history. Okay? And why it's also really important to look at a number of different genetic markers, for example, if you want to understand a whole history. You can't just look at mitochondrial DNA, you can't just look at Y chromosome DNA, and in many cases, other researchers are using nuclear DNA to address other questions. So when we're looking at DNA, whether it's mitochondrial or Y chromosome, and we're looking at trying to understand evolutionary relationships, what we're talking about is the DNA sequence. And you'll hear us talking about comparing sequences, and that is the sequence of bases of nucleotides, okay? And so we talk about DNA sequences and base pairs, and how many base pairs of DNA we looked at, okay? And that's based on, we'll only talk about one of the DNA sequences, but of course we know what is on the other side, the other strand of the DNA. Okay? So you'll hear us referring to these terms quite regularly. Now, what we're looking at in terms of trying to understand or compare DNA sequences across space, across the world, and through time, is we're looking at variation and trying to understand how that variation occurred and when those mutations might have occurred. And it's mutations or changes or differences in the DNA sequences between individuals and those differences that we see in different populations that help us reconstruct the evolutionary history of our species, or of any species, really, the history of life. So whether we're looking at DNA between species or within a species. So the mutations that we're normally looking at in terms of the DNA, of mitochondrial DNA and Y chromosome markers, are single nucleotide polymorphisms, or single base changes. We sometimes refer to these as SNPs. Okay? These are sites where we tend to see a lot of mutation. Okay? So we're looking at SNPs often and mutations, but mutations can be of a number of different types. Okay? So we have on the top there on the slide the normal, okay, mitochondria, or normal DNA sequence. In this case, it's CAT, CAT, CAT. You can have a SNP, a single base change, in which an A mutates to a C. Okay, so instead of CAT, 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 you now have CAT, C, CT, CAT. Okay, so that's a single base change. You can also have what we call insertions or deletions, or the addition or deletion of DNA of a single base or multiple bases. In this case, we have the insertion or addition of 2 Gs here, okay, comparing the normal to this third type of DNA. Okay, so that's an insertion or an addition mutation. Or you can have deletions, and again, they can be single bases, or in many cases, very large chunks of DNA that are deleted or inserted. So here we have a deletion. Okay, deletion of the T there. So it reads, instead of CAT, it reads CAC. Okay? So those are the different kinds of mutations that we use in order to try to compare DNA sequences, or to try to understand and recreate the evolutionary relationships. So as an example, we'll take DNA sequence from one individual. Okay? We can then compare that to sample 2 from another individual. And we align the DNA sequences so that we're looking at the same bit of DNA. And we can see here from this alignment, comparing sample 1 to sample 2, there's one difference there, one mutation. That's a SNP, from a T to a C. Okay? We can then align another sample and compare all 3 of those samples. And when we align those 3, we see that sample 3 has the same C mutation, okay, that we saw in sample 2. It makes it different from sample 1. But it also has another mutation, okay, and that's a change, another single base change, from a T to an A. We then compare sample 4. And we see, again, sample 4 has the same C mutation that we saw in samples 2 and 3. It has the same A mutation that we saw in sample 3, but it also has a different mutation, a mutation from a C to a G. So we can, by comparing these, we see how closely related the different sequences are, and we can start to reconstruct a phylogeny, or an evolutionary tree, a whakapapa, you might say, of DNA evolution. Okay? And if you remember back to a previous seminar that Hamish and Crid did on kelp, and Hamish gave a very nice discussion about phylogenies and the development of phylogenetic trees and how we can do this. So very simple when you have 4 samples and just a few bases, but when you're actually looking at a huge amount of DNA sequence, we use computers to do this. But simply, we can look at sample 1 and sample 2, separated by a single base difference. They're more closely related than sample 3 to sample 1, separated by 2 bases, and sample 4 is the most different from sample 1. So we can create a very, very simple kind of phylogenetic tree, but we can develop this for more sequence data and more samples, and incorporate lots of different evolutionary models and things. So go back and have a look at Hamish and Crid's discussion of phylogenetic trees. So getting back to the settlement of the Pacific, one of the really interesting things, in particular the situation in terms of the settlement of Polynesia, as Michael said, it was one of the most recent settlement events in human history. It was the last region that was settled by modern human populations. And so as a result, we've actually got amazing information about the history and the whakapapa of the people in the Pacific. And people can identify their evolutionary history. Where did they come from? Okay? We can trace it back to a particular waka. We can trace it back to Hawaii. But we're interested in what about further back? What about more kind of ultimate origins of populations? What are the relationships between all of the different Pacific populations? And what are the relationships between the Pacific populations and the rest of the people in the world? How does this relate to the out of Africa kind of migration? So Marshall's now going to talk a little bit about the Pacific and how we see the Pacific.
Okay. You've probably all seen maps with these three terms on them: Melanesia, Polynesia and Micronesia. But I wonder if you're aware of the history of where the terms come from. They actually are the names that were given to different geographic regions of the Pacific by the first European explorers. And those explorers divided up the Pacific into these three regions, basically based on factors like geography, looking at the people that were living there. And those terms are very much stuck in our usage today. You still hear them referred to a lot. But what we're increasingly starting to understand is that those terms, as a way of dividing up or classifying people, actually have no biological or scientific basis to them. So that Melanesia versus Polynesia, the differences between the populations are not the lines dividing them are not necessarily the lines that we look at, particularly once we start to look at mitochondrial DNA and Y chromosome DNA. So for that reason, scientists working in this area have divided the Pacific up in a slightly different way. And the line shown here on the map divides the two main areas, and they're divided primarily based on age of settlement. On the left-hand side of the line, we have Near Oceania, and that's the oldest settled area of the Pacific region. On the right-hand side of the line, we have Remote Oceania, and we know that that's the area that was most recently settled, and most probably in the last 2,000 to 3,500 years. So when we talk about a lot of this stuff, we don't tend to refer to Polynesia, when scientists talk about a lot of this sort of thing, they don't tend to use Polynesia, Melanesia, Micronesia, because the data doesn't match with it. So based on that and keeping that in mind, Lisa's now going to take you through what we know, particularly about what happened in Remote Oceania, and then we're going to what we know about what happened in Near Oceania, and then we're going to move through into what we think is happening in Remote Oceania.
Okay, it's really important when we're asking questions about prehistory and what happened in the past that we think about the fact and we realize the fact that the environment looked quite different to what it does today. So we're talking tens of thousands of years ago. Okay, we've had, ice ages and things happening in between now and then. And at the time that people first came into the Pacific region, okay, the landscape looked quite different. So at the time that people came in, 40, 50, 60,000 years ago, okay, sea levels were much, much lower than they are today. Okay? That means the islands that we now think of as island Southeast Asia were actually joined to the mainland of Asia into a larger Asian continent we refer to as Sunda. Okay? Australia, New Guinea, Tasmania, again, all joined together into a large land mass we refer to as Sahul. Okay? But there are actually deep water trenches that exist between Sunda and Sahul, okay, that were never resulted in a land bridge. Okay, so Sunda and Sahul were never connected. But when people left Africa 90,000 years ago, traveling along the south coast, they could walk all the way through what is now island Southeast Asia to the edge of Wallacea. We know that biogeographic boundary of Wallace's line, and that separates your placental from your marsupial mammals, and you have very, very different biogeography between the Asian and the Australian flora and fauna. Well, people could walk through Sunda, but they had to get across those water gaps, okay, in order to reach Sahul, in order to reach Australia and New Guinea. And they did this at least 50,000 years ago. Now, this required some kind of watercraft, and this is the earliest evidence around the world of people moving across large water gaps. We're talking gaps of 70 kilometers. This is not something that somebody could swim across. So people used some form of watercraft. We don't have any archaeological evidence of that. But they reached Sahul, and they moved through that land mass of Sahul, which is now Australia and New Guinea, very, very quickly. It looks like multiple populations arrived. It wasn't the single pregnant woman, holding onto a log, being washed ashore and landing on the shores of Australia. No, there were multiple population migrations or multiple populations coming through. And they very, very quickly settled the entire continent. But these aren't large populations. They're relatively small populations that become isolated. We're talking tens of thousands of years ago, so a lot of time for variation to occur between populations that aren't mixing a lot. Okay, so we see incredible genetic and linguistic diversity in Sahul. Okay, long period of time and relatively isolated populations. But we know from the archaeological evidence that people got all the way around the continent. We have people down in southern Australia 45,000 years ago, archaeological evidence of this. Okay? But basically hunter-gatherer populations. But they're adapting to their environment, okay, in numerous ways. We have some of the earliest evidence of agriculture, independent development of agriculture in the New Guinea highlands, okay, as early as it's happening elsewhere in the world. Okay, so people are adapting and modifying their environment and so forth. But we see a significant change happening in this region of Sahul at about 3,500 years ago. 3,500 years ago, we see the appearance in the archaeological record of a new type of settlement. All of a sudden, people are not necessarily kind of using the resources around them in relatively mobile populations. We have the establishment of villages. And what we see in the archaeological record on the coast of New Guinea, for example, on the islands in the Bismarck Archipelago, are villages, stilt villages, houses, like you see in many places of the Pacific today, houses being built out over the reef flats. And we see the house posts in the archaeological sites. We see the appearance of a number of new plants and animals in the archaeological record, and we see the appearance of pottery. And this pottery, and in particular the designs of the pottery, you see these very, very intricate dentate stamped, or these kind of comb-like designs that are being imprinted into the pottery, we refer to as lapita pottery. And this whole kind of complex of new artifacts that turn up and these new settlement patterns and things, we identify archaeologically as the lapita cultural complex. And it looks like this is a new kind of a movement in of a new people into the Pacific region that might be associated with the whole Neolithic expansion. As populations grow with development of rice agriculture, for example, populations expand and start moving into new areas. So what we see archaeologically is this dispersal into Near Oceania, where people have been for 40,000 years, okay, along through the Bismarck Archipelago. We see the first lapita sites appearing in about 3,350 years before present, or years BP. Okay? They move quickly through Near Oceania and break through that boundary into remote Oceania. Now, when they move into remote Oceania, the islands of the Reef Santa Cruz or Southeast Solomon Islands and eastwards, they're the first people to arrive on these islands. These are pristine islands where nobody has ever been before. Okay? So very, very quickly, they move out into remote Oceania, reaching the Reef Santa Cruz Islands about 3,100 years ago. Vanuatu, New Caledonia are settled almost simultaneously, and by 2,900 years ago, they reach the edge of the Polynesian Triangle. We see lapita sites in Samoa and Tonga. Okay? For some reason, lapita expansion stops. We don't know why it stops there. But it's in this kind of homeland region, in this lapita, out of lapita populations in Samoa and Tonga and eastern Fiji, that we believe ancestral, that's the Hawaiki in a sense, where ancestral Polynesian language, society, and culture develops over a period of time. And after maybe 1,000, 1,500 years, we see populations moving out of Samoa and Tonga to the rest of the Polynesian Triangle, going out to the Society Islands, Tahiti, and the Cook Islands, but ultimately up to Hawaii by about 1,200 years BP, years ago. Out to Easter Island, maybe 1,000, between 800 and 1,200 years BP, or years before present. And ultimately down here to Aotearoa, New Zealand, about 700 or 800 years ago. And from here, probably over to the Chatham Islands about 500 years ago. Okay? So that explains, in a sense, from the archaeological perspective, what we know about Polynesian settlement. But how do we actually know this? What is the other evidence that we have, okay, for this migration or the origins and tracking the origins? Well, linguists actually have been looking at similarities between languages. And the first Europeans to arrive in the Pacific were remarked on how similar the languages were and how Polynesians could communicate with one another, and the similarities between generally between the languages of the Pacific. And that's because all of the languages of the people that they were speaking to belong to the Austronesian language family. They belong to the same general language group. Okay? Now, linguists have been able to track the origin of Austronesian languages back to Taiwan. Okay? So if you start looking at Polynesian origins and do a Google search, you'll often see this link. Polynesians came from Taiwan. Okay? This is based on a linguistic model, based on the Austronesian languages, and being able to reconstruct the history of the languages, and that's clearly linked to the history of the dispersal of Austronesian-speaking people. And so what we see is that the Austronesian languages started in Taiwan. That's where they emerged. And we think people left Taiwan probably around 6,000 years ago. As I say, many people are linking it with this Neolithic expansion out of mainland Asia into Taiwan and then down through island Southeast Asia. Okay, and we see the language starts to break up. We see particular language families being identified. A Western Malayo-Polynesian group of languages spoken in parts of island Southeast Asia. We then see people continuing to move towards the Pacific, and we see the Central Malaysian Polynesian languages being spoken, and ultimately out in remote Oceania, in the Oceania generally, we see the people speaking the oceanic subgroup of Austronesian languages. So we can actually reconstruct language history in a very similar way that we reconstruct DNA phylogenies. And so there's some really interesting work that's being done, again, here in New Zealand, researchers here at the University of Auckland, on the evolution of the Austronesian languages and applying biological tools and the computer programs that we've designed for DNA to language differences and looking at the history of Austronesian languages. But what about the evidence from DNA, okay, from mitochondrial and Y chromosome data? Well, again, let's go back and start thinking about out of Africa. We can compare all of the DNA sequences from people around the world and start to see patterns that suggest the movement of people through time and across space. And so the most diverse lineages that we see in mitochondrial DNA, and this is based on the work of Alan Wilson and his students, are the lineages that we find in Africa. And these lineages were initially given, they were called lineages 1, 2, and 3, and then they were just given letters, L1, L2, L3. And the lineages that diverged from these or came out of these lineages were given new letters and numeric kind of combinations so that we could start to understand the evolutionary history. So the L1 and L2 and L3 lineages we find only in Africa. Okay? But it was the L3 lineage that we think was the out of Africa, that first out of Africa migration. That was the group of people that went out. And the L3 lineage splits into two branches, an M branch and an N branch. Okay? When we look at the distribution of lineages that belong to these two M and N branches, we start to see some interesting patterns. For example, the M lineage, and those derived from the M branch, are only found in Asian populations and Asian-derived populations. Okay? The lineages that we see on the N branch, and N splits into an R lineage as well, which breaks down further, we see both in Europe and in Asia. So that tells us something about the populations that are moving. One goes exclusively to Asia, and one splits, and some people go to Asia and some people go to Africa. Now we can start to look at very specific lineages further down in the tree, and we notice that some of these are only found in Pacific populations. Okay? These Q lineages, P lineages, and some of these very ancient M lineages, like this one's called M28. These are lineages that are found only in the Pacific region, amongst Pacific populations. And when they calculate the emergence of these lineages, when did the mutations that define these lineages occur, we estimate that they're very, very ancient. And they occurred at least 50,000 years ago, and they're exclusive to the Pacific. So this fits very nicely with the archaeological evidence, which suggests that these people came through into Sahul 45, 50,000 years ago. Okay? But we also have other lineages in the Pacific. at high frequency in particular populations. And in particular, we're going to be talking about the B4 lineages and lineages derived from the B4. So we see that break down further into B4A and B4A1 and B4A1A1. And so we can kind of start to understand the evolution by looking at the mutations within the B4 lineages. Now, these B4 lineages arrived much more recently. They're very recent arrival in the Pacific, and the age of the B4 lineage is less than 10,000 years. Okay. So again, we have a story about archaeology and Lapita arrives about 3,500 years ago in the Pacific, and we see spread of Austronesian languages. Okay, so these stories, and then we have these B4 lineages that are arriving, maybe part of, again, this Neolithic expansion. So we can start to look at the distribution of these different lineages across the Pacific and think about how these different populations are moving. So what we see when we look at these lineages across the Pacific is that in remote Oceania, and in Polynesia in particular, we find very, very high frequencies, almost 100% frequency in some East Polynesian populations of these B4 lineages. Okay? And we see on the top of this slide, the yellow and orange colored pies, those are the Asian derived, the more recent derived mitochondrial DNA lineages. And down below that, we see the Near-Oceanic, these ancient P and Q and M lineages. Okay? And you can see in Polynesia, we see very high frequency of Asian derived mitochondrial lineages and much lower frequency of Near-Oceanic derived, those ancient lineages. So again, seems to fit very, very nicely with the story that's emerging out of archaeology and linguistics, that you have this migration out of Asia through Near-Oceania and into remote Oceania. And they're picking up some of these Near-Oceanic mitochondrial, ancient mitochondrial lineages as they go, but at very, very low frequency. But then people started looking at the Y chromosome, and technology developed so that we could start looking at the Y chromosome variation. And so it came slightly after the mitochondrial DNA data was emerging. And all of a sudden, we started getting a very different picture with the Y chromosome. Okay? So with the Y chromosome, we see that there are some Asian Y chromosome lineages that we can trace back to a recent migration out of Asia. But a very large proportion of the Y chromosomes that we see in the Pacific are Near-Oceanic. Okay? So the more data we accumulate from different parts of the genome or different parts of different markers, different genetic markers, we might be getting different stories. Okay? And how can that be? Well, men and women behave differently. Okay? So this has been very, very interesting in terms of trying to understand when you start comparing mitochondrial DNA variation and patterns that we see that might suggest one history, we can see a very different history when we start looking at another marker, in this case, the Y chromosome marker, which suggests that there was much more mixture along the male line than there is along the female line. So you can start to think, how might this occur? Okay? Why might we see different patterns? We often think, well, who are your voyagers? Who are your explorers? As an interesting comparison, we see a lot more European Y chromosome markers in Pacific populations than we see European mitochondrial markers. So you think of who were the whalers and the sealers and the sailors and the explorers, a lot of European males kind of coming through and spreading their Y chromosomes around the world, kind of more at higher frequency than mitochondrial DNA would be spreading. So there's some problems with trying to compare variation that we see in modern populations. We know populations move around. We look at the variation, the biological variation we see in this room. We're all sitting here in Aotearoa. Okay, we're sitting here, and yet we all have different origins, maybe spoke different languages. People move. People move today. People moved in the past. Okay? We have to think about what is the population genetics, what's the history that's going on between the first people that arrived, and that's the question we're trying to understand, and the variation that we see today. What are the various factors? So, admixture, clearly one of the things that we have to think about. Both historic admixture, people moving around recently with airplanes and ships and things, but also prehistorically. These people could get in canoes and move around all the time. Okay? So this blurs the picture when you're trying to understand prehistory and you're using modern human variation to try to answer the question. Okay? Founder effects, and we'll talk about this in a minute. Who was in the canoe? Okay? How representative of the population, the founding population, is it representative of the source population from which it came? So this is something we have to consider when we're looking at any island settlement, whether it's humans or animals. What about population bottlenecks, natural selection? The introduction of European diseases had a huge impact on many Pacific populations. Huge population crashes. In some cases, you lost, over 50% of the population. So how does that affect what we see today and what we're inferring about the past? There are a number of ways we can get around these problems of trying to figure out, based solely on what we see today, what happened in the past. And the use of ancient DNA is one of those. And that's something that has been very much dependent on the development of new technology. The ability to get DNA out of ancient remains is something relatively recent. And that technology is changing so quickly. We've just had another major revolution with the introduction of next generation sequencing, which you guys might be hearing about in technology courses and things. So when you actually go to ancient DNA, you're getting DNA from those actual people dated to an actual point in time. We can get radiocarbon date and DNA sequence out of the same ancient DNA sample. So we're not guessing what was happening in the past. We're getting the actual tangible DNA of those individuals. Okay? And we can find that DNA in plants and animals and people, human remains. It also, so that removes the issue of modern admixture. Okay? But there's also another approach, and this is an approach that we took really as a result of ethical issues of dealing with human tissue and human remains, and trying to basically work with local communities at doing DNA, of developing kind of a proxy for tracking human migration by using human DNA and trying to get at it in another way. So Marshall's going to talk to you a little bit about commensal approaches.
Thank you. Okay. The commensal model. Let's just look a few minutes at this word commensal. Now, it'll ring bells with a lot of you from Year 12 biology when you talked about commensalism as an example of an interspecific relationship. And the words are related, but the way in which it's been used here, when we're looking at this idea of movement of people, migration of peoples, the word has a slightly different meaning, and commensal literally means to eat at the same table. So the commensal model relies on looking at organisms that have an incredibly close relationship with humans. So close that they can be used, their movement can be used to model the movement of the humans with which they're associated. And one of the best examples of a model animal used this way is the coyote or the Pacific rat, otherwise known as Rattus exulans. Now, it's not the only commensal animal. Scientists have also looked at the dog, the chickens, and the pig. Now, what made these animals so good as commensal models was that they all travelled in the canoes with those early Pacific people as they set out to explore the Pacific. So they were taken along intentionally, and they were all there for one reason. They were a food source. Every single one of those animals was a food source. So you think about those early Pacific peoples, they're setting off a long journey over water. They don't know exactly when they're going to find land. They need to have food with them. So they took their food to make sure that when they got to the other end, that there was something for them. So by looking at these animals and doing exactly the same sort of analysis that Lisa's been talking about with the human DNA, in particular the mitochondrial DNA, looking at the mitochondrial DNA of the modern populations, we can actually use them to model what the humans were doing at the same time. Now, the coyote is particularly powerful as a model because it hasn't, the modern coyote hasn't interbred with the other rats that were introduced when the Europeans arrived. That's the Norwegian rat and the ship rat. So the mitochondrial DNA of the modern coyote population hasn't had that problem of admixture with modern populations. We can't say the same though for the dog, the pig and the chicken, so that was something that Lisa and her team had to take into account when they looked at it, but there are ways around it. So Lisa's actually going to tell you now specifically about the work that she did with the rat and what she found out about it. And this work, when it was published, was so significant that it actually made the cover page of a very important journal in April 2004. And this was a journal called The Proceedings of the National Academy of Science. And the picture that you see there is from the cover of that journal, and Lisa's going to tell you why she put that picture on the cover.
Now this is actually from a carving from the University of Whakana Marae. So if you go down to the Parewhakairo, you can actually see this carving. And this is a carving of Ruanui, the navigator, with a kiore traveling on his shoulder, okay, as a traveling companion. So very, very different relationship with Pacific people and rats than we often think of rats as being bad and dirty and evil. These are a traveling companion, a really important part of Pacific people's transported landscapes that they took with them when they arrived on the islands. So because the kiora travel so closely with humans, they work as a proxy for the movement of humans. They can't get around on their own. They couldn't get to Aotearoa on their own. They had to be brought in the canoes. So you figure out where the kiore in New Zealand came from, and that tells you where the waka came from, the colonizing canoes. And we can trace that all the way back through the Pacific. So we collected rats from all around the world, from archaeological sites, and we went trapping fresh populations. So we could compare ancient DNA and modern DNA. And we were really surprised. We extracted DNA, mitochondrial DNA, amplified, again, a variable region of the mitochondrial genome, and compared sequences of rats from all the way across the Pacific and island Southeast Asia. And we found 3 very distinct groups of DNA lineages. And we identified these as group 1, group 2, and group 3. And when we looked at the map, we actually were very, very surprised at how well they patterned kind of biogeographically on the map. So you could see the group 1 kiore, okay, we found only in island Southeast Asia. Okay, we didn't find any of those lineages anywhere else in the Pacific. The group 2 rats we found in island Southeast Asia and moving all the way through near Oceania, okay, to the edge of remote Oceania. Okay. The group 3 rats we found exclusively in remote Oceania, and the only other place that we saw them was in Halmahera, which is in Wallacea, okay, an island in Wallacea. But given this model of Lapita, okay, people were moving through near Oceania, island Southeast Asia, through near Oceania and out into remote Oceania, we were really surprised that we didn't see any of the group 3 kiore in near Oceania. We said, this doesn't really fit with the archaeological model and the whole Lapita expansion. We know that there are rats in the earliest archaeological Lapita sites. We find them in the earliest sites in remote Oceania. How are they getting out here? Where are they coming from? And this is a really important point, to go back and challenge your data and say, what do we expect to find and what do we find? And is there some kind of bias in our sampling? Have we really got the right data that allows us to test our hypothesis? When we went back and we looked at this, and we started looking at where do we have samples in near Oceania, we realized we didn't have any DNA samples from actual islands that had evidence of Lapita settlement. Okay? Most of our samples came from the mainland of New Guinea. Okay? And they came from islands in the Solomons where there is no evidence of Lapita. So we thought we actually don't have the data to test this hypothesis, so we better go back. And so we went back into the field and started sampling in near Oceania, in the Bismarck Archipelago, working with archaeologists, looking for archaeological evidence of Lapita and sites where we know that there are Lapita archaeological sites, and we got rats from those sites. And interestingly, we indeed did find that the remote oceanic lineages were on some of those islands. Not all of the islands, which are Lapita sites, okay, but they are found on some of those islands. And I started saying, okay, why on some and why not on others? The islands that we saw, the remote oceanic lineages, were also islands where we had other evidence, linguistic and archaeological evidence, of interaction with Micronesia. And we see these lineages in Micronesia. I thought, okay, well, that's very interesting. Again, makes you scratch your head and go back and rethink, okay, reformulate. So that was really very interesting. We started looking at the DNA of the other animals, the other commensal animals, the animals that were carried in the canoes. What about the dogs and the pigs and the chickens? As Michael explained, we can't use modern DNA to look at those those animals because Europeans brought dogs and pigs and chickens with them that are the same species as those that Pacific people brought out thousands of years ago, okay? And those animals have been interbred. Okay? So we have that whole issue of admixture. So we started looking at archaeological remains of pigs and dogs and chickens. Now the pigs gave us one history. We got one lineage of pigs coming from island Southeast Asia, or coming from probably mainland Asia, Vietnam area, moving through island Southeast Asia and out into the Pacific. There's a single pig lineage. Okay? But what's interesting, the dogs, we found 2 mitochondrial DNA lineages of dogs and ancient dogs from Polynesia. One of those we also found in places like New Guinea, in Neuro-Oceania. But the other one we didn't find in Neuro-Oceania at all. We only found it in Polynesia, and it was found in a modern Indonesian dog. Okay, so we have it in island Southeast Asia, and we have it out in Polynesia. I thought, well, that's very interesting. We then started looking at the chicken DNA. So we find chickens in early Lapita sites, and we compared the DNA across the Pacific from ancient remains, and we actually have now found Pacific chickens in South America, but that's a whole other story. But when we looked at the chicken DNA, we again saw 2 lineages in Polynesia. One that looks like it was coming through remote Oceania, or near Oceania into remote Oceania, but then a second lineage that was coming through Micronesia, from island Southeast Asia, through Micronesia, and into Polynesia. So we're getting some very interesting patterns when we start looking at the animal DNA, ancient DNA, modern DNA, and we started thinking, well, how might this fit with the human DNA? One of the explanations for this very, very high frequency of these Asian mitochondrial markers is that it's been a population bottleneck, okay, a founder effect in a sense, where the population that went out into Polynesia from Neuro-Oceania through Vanuatu, New Caledonia, and out into Polynesia was not representative of the population from which it came. It was just a subset. And that's been the explanation for this high frequency of Asian mitochondrial DNA in Polynesia that we don't see in places like Vanuatu or the Solomon Islands. We do see some of the lineages there, but not at such high frequency. We suggested, based on the animal DNA, there might also be another model that we can actually start to test now, and that maybe the history of the Pacific isn't as simple as this model that we developed from archaeology and linguistics. And maybe we're missing some later movement of people. So we have Lapita people coming through to Samoa and Tonga, but then maybe 1,500 years ago, okay, we see another population coming out of Asia through Micronesia and into West Polynesia, mixing with Lapita people, and that might be the thing that pushes then people out into settling the rest of the Polynesian triangle. So this is something that we can actually start testing now with ancient and modern DNA of the animals. And as populations are more interested, as indigenous communities are more interested in participating in this research, we can start looking at ancient DNA and modern DNA of humans as well across the Pacific in a very specific way to start answering some of these questions. So, Michael.
Now, Lisa's talked a lot about the founder effect as one explanation for why some of this DNA evidence doesn't all quite fit together. So we're just going to do a quick diversion for a moment to talk about, well, what exactly is the founder effect? So what we have here is an island population. It's been established for a while. There's reasonable genetic diversity on it. Part of that population decides, after a lot of planning and thought, that they want to go exploring and see if they can find a new island. So they get in a canoe and they move to a new island. Now note those people on the canoe. Look at their genetic diversity. When they get to the new island, they move on, it suits them, so they decide to stay and they establish themselves as a population. Of course, that canoe, to do that, would have had to consist of both males and females. So over time, their population grows. But compare the variation in that population with the variation back in the original one. You'll see that it's a lot less in the new population. And this is due solely to the fact that that founding population, the group of people in the canoe, their genetic variation was not representative of the original population. And that at its heart is what the founder effect is about. When a new population is established and the establishing group doesn't genetically represent the original population. Now, there's been quite a lot of research done in this area, including some work done by scientists here at New Zealand looking at genetic variation in New Zealand Māori, and Lisa's going to tell you a little bit about what they found out and what it might mean.
Now, generally, founder effect can affect a population in numerous ways. It can either increase genetic diversity or decrease genetic diversity. And when you have serial founder effects, or settlement from one island to the next, variation can change. But what we see in the Pacific, it looks like we have a decrease in mitochondrial DNA diversity as we move across the Pacific. And this is something that researchers, again, here in New Zealand, looking at mitochondrial DNA variation in Māori, have found is that there's very limited mitochondrial DNA variation in Māori. This may mean that the founding population may have been closely related women, okay? A family-based settlement, for example. Okay? Or, again, we don't know how well the modern variation represents the variation in the past. But this is one possible explanation. So when we looked at those B4 lineages, we see that nearly 100% or 98% of the mitochondrial DNA lineages that we see in New Zealand, okay, in Māori, belong to those B4 lineages. Okay. Limited number of lineages that we see. The lineages that we see, and in this early study they identified 4 main mitochondrial lineages, are a subset of the variation that was seen in East Polynesia, okay, which again was a subset of the variation that they saw in West Polynesia, which again was a subset. There wasn't as much variation in West Polynesia or in Polynesia generally as we see in the rest of the Pacific and Melanesia. And so you can see, in a sense, there's reducing variation as you move across the Pacific. So one of the things that we're actually doing now is working with Pacific communities. And I'm working with National Geographic and their Genographic project. I'm trying to understand the genetic variation that we see in the Pacific, working with Pacific Island communities to sample, not from blood samples, but from cheek swabs, taking DNA from cells just scraped from the side of the cheek, working with communities often who are interested in the ancient DNA from their homelands as well, to try to understand and ask very specific questions about the settlement of the Pacific and the history of populations kind of through time and space in the Pacific region. So, Maisha?
Right. Okay, so that's our story of what we currently understand as a model for the settlement of the Pacific. So what we want you all to do now is to go away and to think about those concepts that I listed right back at the beginning of the seminar. How did they relate to what you've heard about Lisa's research and some of the research from other scientists, and apply that to our challenge questions. So we've got 4 challenge questions for you today. First one's a very straightforward one. We've got a couple that are working at probably about excellence to just under scholarship level, and then we've got a couple that are at scholarship level for you. So this first one very much relates to what we've just been talking about, Lisa and me. The idea of what's the impact of settlement by small groups of people on genetic diversity within Polynesian populations. The second one, again, not too difficult, you will need to do some thinking. Why is it that we tend to use mitochondrial DNA rather than nuclear DNA in research like this? And what are the differences in information that we can get between ancient DNA and modern mitochondrial DNA samples? Now, the next challenge is starting to work at scholarship level, because what we want you to do is take your knowledge of biotechnology, and we've talked a lot about the genome analysis as it applies to mitochondrial DNA and Y chromosomes. How has this advanced the ability of scientists to investigate the hypothesis that there was more than one wave of migration into the Pacific? And finally, looking at the use of the commensal animals. First up, what is a commensal animal? How can they be used to study it? And then the challenge part of the question. What are the ethical and biological implications of using model species for looking at the migration of the Pacific as an alternative to using human samples? Now here, when we talk about ethical and biological, we want you to look at those 2 ideas in relationship of both the animal and also the use of human samples. So remember, Talk about it in your post-seminar workshops next week with your friends. Have a go at answering the questions, and we'd love to see you posting it on the wiki. So what we're going to go to now is the questions, and we'll start with a question that came in before the seminar, and then we're going to cross to the seminar chat, where I gather there's been a reasonably interesting discussion going on. So Lisa, if you'd like to come up and join me. So our first question actually comes from a Year 10 student at Kelston Boys High School, and he was asking, how does ancient DNA remain stable? Lisa?
Well, interestingly, ancient DNA doesn't remain stable. As soon as the organism dies, the DNA starts breaking down. And this presents one of the major problems with ancient DNA, is that it's generally very degraded. And it's degraded based on age of the sample, how old it is, okay, but also on what's the location of the sample, okay, the context of the sample. So an archaeological sample that is in permafrost, okay, is preserved very well. And people have found, looking at penguin DNA from the Antarctic, they're getting beautiful DNA out of this nicely frozen sample. Okay. Samples in the Pacific, okay, where they're out in an archaeological site in the Pacific that's hot and wet. Hot and wet are the worst conditions you can have for DNA preservation. Okay? Water percolating through the site, all of that is breaking down DNA at a fast rate. So what we find in the Pacific is that the conditions are generally pretty bad. Cave sites are great. Neanderthal, some of you may have heard about the Neanderthal genome sequencing. Those are samples that are 40,000 years old, okay? And they're getting DNA out of the Neanderthal remains. Those are in dry cave sites, for example. But the other thing that's really important about ancient DNA, and because of this whole breakdown of the DNA and the way it degrades into short fragments and you get kind of mutation events that chemically the DNA is changed, and we know how that happens and what it kind of looks like. But what's really exciting is the new technology, this next generation sequencing. Okay? It allows us to actually get at single molecules of DNA and amplify DNA from these little tiny pieces. It's perfect for ancient DNA. It allows us to amplify small fragments from DNA that in the past we couldn't have gotten from samples that we couldn't have gotten DNA out of in the past. We can do this now with the new technology. And this has just happened in the last few years. And that's what enabled the whole Neanderthal genome project to be developed, as this new next generation sequencing. So we've got this technology here in New Zealand. In fact, some of the best people doing some of this work, and we're applying this to a lot of questions, not only in terms of human evolution, but in terms of animal evolution generally. So it doesn't remain stable. It does break down through time. Once you get to a fossil situation, when a sample is fossilized, that means there's no organic material in it. You can't get DNA out of it. So you're working with kind of time and conditions and preservation, but it does break down. It lasts for thousands of years, and in some cases tens of thousands of years. But that depends on where the sample is found.
Okay. Great, thank you, Lisa. All right, so what we'll do now is we'll go to a question from the seminar chat, and just before we do that, if anybody in the live audience has a question, can you just go and stand behind the microphone, and I'll get you to ask your question after we've had the seminar chat. Okay, Anna, first question.
Hi, Michelle. We've got a question. How long does the founder effect last in a population the size of most Pacific islands? Is the reduction in variation in the gene pool recovered after a number of generations, or is it impossible to recover without immigration?
Very good question. Generally, in terms of a founder effect, what you start out with is what you're working from, your base that you're working from. So unless you have immigration, then it's highly unlikely that you're going to regenerate the variation from the original population. However, over long periods of time, new mutations are likely to occur. So you can actually, have an increase in genetic variation of population through time. But again, how much time is, not very long period of time. We're talking about the settlement of the Pacific, only a few thousand years, versus tens of thousand years, you can actually accumulate more variation. So, but certainly we do have to think about populations, the movement of people, and migration and immigration, and in all of our modeling of understanding and explaining the variation that we see around the world.
Okay, great, thank you. Well, so far nobody from the live audience, so Anna, another question from you, please.
Yep, we have another question. How is mtDNA extracted in the way that it is separate? Sorry, let me start again. How is the mtDNA extracted in a way that is separate from nuclear DNA?
Good question.
Very good question, and I'm very pleased to say that, again, biotechnology has been fantastic for us, because in the past, when Alan Wilson was doing this work, they did actually have to extract mitochondrial DNA using cesium chloride gradients, and it was a very complicated process. Now, we use PCR to actually target the mitochondrial DNA. So we usually extract total genomic DNA, all the DNA of the sample, and then we use PCR, the polymerase chain reaction, to actually target the mitochondrial DNA, if that's what we're interested in, and amplify that DNA to the point that we actually have enough of it to sequence and analyze. So we don't extract the mitochondrial DNA, we just preferentially amplify it using the PCR process, or the polymerase chain reaction.
Lisa, is that due to the fact that mitochondrial DNA is much smaller than generally much smaller than nuclear DNA?
No.
Okay.
The fact, no.
That's fine. It's a lot more complicated than that.
It is a lot more complicated than that. It's just, I mean, with PCR you can amplify, you can target any particular part of the genome that you're interested in. We can, in terms of ancient DNA, in the past, we tended to focus on mitochondrial DNA because of preservation issues and of copy number issues. So it used to be that when we looked at ancient DNA, because you have more copies of the mitochondrial genome in every cell than you have of the nuclear genome, okay, you have a better chance of it being preserved in an archaeological or an ancient DNA sample. So you've only got one copy of nuclear DNA in each cell, but you've got multiple copies of the mitochondrial DNA. So in the past, ancient DNA was really limited to doing mitochondrial DNA research, or if you had very well-preserved samples, you could sometimes get some Y chromosome data out of it. But again, with a new next-generation sequencing technology, you actually are able to go beyond the mitochondrial DNA and deal with very, very tiny amounts of nuclear DNA and amplify those so that you can analyze it. So again, things are changing dramatically in the last couple of years. It introduces us to the world of genomics. It introduces us to a lot of new possibilities for ancient DNA research as well. So it's a very, very exciting time.
Good. Thank you very much. Well, nobody from the live audience yet. Throwing open a challenge. Let's not have the seminar go by without a question from the live audience. Okay, Anna, another question from you.
Hi again. The information that this work generates is very interesting in terms of looking back at our history. How does this information impact on modern populations, and can it be used to explore aspects of social well-being today?
Again, excellent question. While as historians we're interested in understanding our past, and I think this always helps us to understand relationships and how closely related we as all humans are, I think a very exciting field that we're hoping to really develop, again, is trying to understand why is it that we see particular diseases, which we know have a genetic component, why do they affect certain populations at a higher frequency or higher rate than we see in other populations? And we see some of those diseases affecting Pacific populations at substantially greater rates than non-Pacific populations. And so by understanding the history and looking at some of these, possibly looking at some of these genetic markers in the ancient remains, it'll help us maybe understand why we have some of these high frequencies of diseases in Pacific populations. And we can start to say, is this a result of, for example, natural selection with the introduction of European diseases that has selected for particular genotypes in a population. So it just helps us understand and tease out the whole nature-nurture component, or environment versus genes, and understanding our history and potentially, help us address some of those health issues in better ways. So, as I say, it's a good question, and it's something I think that we're really starting to think about now that, again, technology is allowing us to maybe get at some of these questions about the past.
Great, thank you, Lisa. And the live audience has responded to my challenge.
They're alive.
Yes. Can you introduce yourself and then ask your question, please?
I'm Nick from Onehunga High School. Could you tell us how human interbreeding with Neanderthals relates to the out of Africa theory?
Again, very good question, and another very good example of how looking at different genetic markers has given us a different perspective on that whole question of interbreeding between Neanderthals and anatomically modern humans, Homo sapiens sapiens, and are they a different species or not? Because when they first started looking at the Neanderthal ancient DNA, they were only looking at mitochondrial DNA. And so what they found when they were looking at the mitochondrial DNA was there was no link between the Neanderthal mitochondrial DNA and anatomically modern human DNA, both either modern populations, but then they went and actually looked at 40,000-year-old Homo sapiens sapiens ancient DNA, and they found that they were clearly distinct. And so that's what led to, again, suggestion they're not a subspecies, but a separate species. But when they actually, using the new technology, were able to get at the nuclear DNA, it looks like there was probably about a 4% contribution of Neanderthal DNA to the modern human gene pool. So by using nuclear DNA and getting at that with using the ancient DNA techniques, we now know that there was some admixture, but it was relatively limited. And if you think about when that admixture was likely to have occurred, and they've been able to look at different Neanderthal populations, it's occurring probably, it's after populations have come out of Africa. So in a sense, it's admixture, but in a specific geographic, I suppose, location.
Great. Thank you, Nick. Anna, another question from the chat, please.
Yes, if current mtDNA in Māori is lacking in diversity, does that not indicate that the originating population was even narrower in terms of mtDNA variation than the current population?
That's an interesting question. Again, it has to do, if you think about the time frame that we're talking about the settlement of the Pacific, you would think that over long periods of time that you would have increasing genetic variation, through time. But we're talking about only a period of about 700 years, and that's not enough time to be accumulating mutations in the mitochondrial genome. So we think that probably there may be, the odd, maybe one mutation or two mutations, but it's not enough time to accumulate significant mutation to significantly change the population. But the question does go back the other way around, and in terms of, was there more variation here in the past, and did the introduction of European diseases or social situations limit the variation through time or reduce the variation through time? So these are questions that we can only get at, really, by comparing ancient and modern DNA.
Great. Some really good questions coming through. We've got one more question from the chat, which means one final opportunity for the live audience, if there's anybody who's got a question and there's somebody responding. Anna, we'll go to you while this gentleman gets himself organised.
Hi, Michelle. What do you think the human DNA evidence will provide that the evidence from the animal model does not provide?
Well, the question is, we don't know. We are seeing different patterns in terms of the different animal models, the different species that we're looking at. But in some cases, as I say, the reason we started the Kiore research, the rat research in the first place, was we were getting around some of the issues of admixture. And we're perhaps seeing more variation in the rat DNA, and so we can tease out some of those questions. If you have very little, if you're starting with a limited amount of variation and you see that it's only a subset of it, you might be missing some particular event, I guess, in time that you might see if you're looking at another genetic marker, or in this case, another species that's traveling with the people. So the thing is, we really don't know until we start looking. And then we've got to combine all of the information from different genetic markers that we can look at, from archaeology, from linguistics, looking at oral traditions, and all of this may be giving us different parts of what is a single story, or a story of a people. So, we, as I said, we can look at our own, histories and say, my own personal history. If you look at my mother's side and my father's side and where I grew up and what the ancient genes are, that I'm carrying, tell you're getting a different story. I'm standing here speaking with an American accent, but I was born in Polynesia and Hawaii, but I'm now living in New Zealand. So, they're all different bits of a story. And by combining all of those lines of evidence and not necessarily assuming one is going to give us the truth, that all of them together are actually the history, then we're going to get a fuller picture of what was going on.
Great, thank you. Right, live audience.
I'll introduce myself.
Yes, please.
I'm Alex from Onehunga High School, and I was wondering what the motivation for the migration was.
Actually, to identify that, but obviously there are a lot of hypotheses. And I think probably one of the obvious ones is it's out there, because it's there. Why did Hillary climb Everest? And basically we have in kind of human migration, people migrate for all kinds of reasons, whether it might be food shortages. And there's no evidence in the Pacific necessarily of a population growth that pushed people in the islands off to a new island. But we think there were probably social factors as well. And if you look at social structure in many islands, chieftainship and position, social position is inherited by the oldest son. Okay, so what does the second son do? Is he always going to be, the number two? Well, if he goes off and sets up, kind of establishes, goes out on his own, the navigator, and says, here's another island we can go to and we can set up, he can be the chief. So we think there may be some social driving factors. There may be, undoubtedly, if we think about tsunami, we all saw that, on the Boxing Day tsunami, what kind of an impact that can have. And people leave. And they might come back later, or they might move to a completely different place. Volcanic eruptions could have a huge impact on people. So it's no doubt a combination of factors, social factors, and also what's out there. What's, we just found another island that's great fishing. There's really great resources. There's some social tensions here. Let's head off to a new place. But what we do know from the archaeological evidence, and it fits, of course, very nicely with oral traditions, is that there was always migrations back and forth, that people could always go home and maintain those kinds of social ties. And we see those social ties, trade and exchange going on, that these aren't isolated populations from the time that they set up. Okay? That there can be interaction, and they can go back, and they're relying on friends and family when resources might get short. They're trading. Sometimes we see islands abandoned and then resettled again later. So it's a dynamic process, and it's a complex process. There are probably multiple things that drove it.
Great, thank you very much. Okay, people, that brings us to the end of this seminar. We had hoped to do a video conference with Otago students at Otago University, but unfortunately technical problems got in the way there. And just an update about the next seminar. The next seminar, which is the last one of the seminar series, is actually the first week of next term. So we do have an overlap with the final week of the work that you're doing on this seminar, because the holidays get in the way. So next week, you will, the update will be letting you know that the seminar paper for that final seminar will be available from our website, so look out for that. And that one, last seminar, ties in really nicely with what Lisa's been talking about, because it's actually focusing on human evolution, but going back further in time. And it does provide a really good summary of what you have learnt in human evolution this year. So those of you who are watching and you know you've got classmates who feel like a quick revision on human evolution, invite them along to the next seminar. So thank you very much. Thank you to all those people in our remote audiences, to the people in the live audience, and we will see you in October. Good night.